a groundbreaking study conducted by researchers at Duke University, has uncovered a significant link between environmental chemical exposure and the development of bladder cancer markers in canines. The research, meticulously detailed in a peer-reviewed publication, involved a novel approach to measuring chemical exposure in dogs. Over 100 dogs participated in the study, each fitted with a silicone device attached to their collar. These devices, designed to passively collect chemicals from the surrounding environment, remained in place for 5 days, providing a comprehensive snapshot of each dog's exposure profile. The analysis focused on a panel of 120 different chemicals, revealing a concerning prevalence of exposure across the canine population. Remarkably, the researchers detected approximately 40 of these chemicals in at least half of the study participants. However, a striking geographical trend emerged. The concentration and diversity of chemicals detected were markedly higher in warmer climates. This observation highlights the complex interplay between environmental factors and chemical exposure, suggesting potential variations in chemical degradation or release depending on ambient temperature. The implications of this research extend far beyond the canine population. Dr. Wise, a key researcher involved in the study, emphasized the significant limitations of the current regulatory framework. Current regulations often assess the toxicity of individual chemicals in isolation, failing to account for the synergistic effects of complex chemical mixtures. The study strongly suggests that the simultaneous exposure to a cocktail of chemicals, a scenario common to both dogs and humans, significantly contributes to the development of bladder cancer and other adverse health outcomes. The researchers concluded that the findings provide compelling evidence for a paradigm shift in chemical regulation, urging a greater focus on evaluating the cumulative effects of multi-chemical exposures. This research calls for a more comprehensive understanding of how different chemicals interact and influence health risks, moving beyond the traditional approach of examining individual chemicals in isolation. The study's findings underscore the pervasive nature of environmental chemical exposure in everyday life. The most likely route of exposure, the researchers suggest, is through dust. Chemicals, readily released from various consumer products, become incorporated into dust particles, which are then inhaled or ingested by animals and humans alike. This highlights the insidious nature of chemical exposure, constantly present in our environments and often invisible to the naked eye. Previous research has already established the potential for dangerous chemicals to leach from plastic dog toys, another significant source of exposure identified in air and water pollution represent further potential sources of exposure, though these pathways were not specifically assessed in the current study, leaving room for future investigations into the broader environmental contributions to chemical exposure. The researchers explicitly acknowledge these limitations, suggesting further research to explore these additional exposure pathways and to expand the scope of chemicals analyzed. The study represents a significant step forward in our understanding of the complex relationship between environmental chemical exposure and the development of bladder cancer, urging a more holistic and preventative approach to safeguarding both animal and human health. The implications of this research are far-reaching and underscore the need for a more comprehensive and integrated strategy in regulating chemical exposure and mitigating its potential health consequence. The urgent need for further research into multi-chemical exposures and the development of more robust regulatory frameworks are undeniable consequences of this important study.